Again, my name is Aaron Sato. I'm the CSO of the Biopharma vertical at Twist Bioscience. It's my pleasure to talk to you today about a case study where we've used one of our antibody phage display libraries that's focused on drugging hard-to-drug GPCR targets using our synthetic antibody phage display libraries. So again, I always say the best companies out there really truly understand the one thing that they're really good at. So for Twist, that's our ability to print DNA. So shown here on this slide is a silicon chip. It's about the same size as a 96 wall plate. And on this chip, we can actually print individual oligos. We can actually print up to 1 million different oligos on this single chip. And those oligos can be up to 300 base pairs in length. So each one of those individual oligos has an explicit sequence. And so this is a fantastic platform to make large pools of individual oligos. And of course, we use this platform to make all kinds of products that I won't talk about today, but again, clonal genes is one of them. And in particular, we can use pools of oligos to make high quality DNA libraries, which is key for the case study that I'll talk about today. So how do we do that? So basically what we can do is we can again take those large pools of oligos, and if you think about an antibody domain, so again, either a heavy chain or a light chain domain, it's basically a collection of 3 different CDR loops in a single germline framework, for example. We can basically make individual pools of oligos that encode for diversity and then stitch those pools of oligos together to make a hypervariable, either heavy chain or light chain domain. So shown here is just one example where we might, for example, have a heavy chain domain library that we're trying to make, where we might make, say, for example, 1,000 oligos that encode for the diversity in CDR1, another 1,000 for CDR2, and maybe 10,000 or more for CDR3. So the reason why making libraries in this way, in my mind, is really game-changing is that, for example, when I synthesize those oligos in those pools, I can make sure that they exactly match the human repertoire. So the resulting antibodies that come out of the library look a lot like you would find in any human antibody derived from a person. I also can remove sequences up front from the synthesis that might lead to issues with isomerization, cleavage, deamidation, or glycosylation, for example. And then finally, and the third advantage, which is actually key to the GPCR library case study that I'll talk about today, is that we actually can encode for specific motifs in those sequences if we choose to. So in Twist Biopharma, we're focused on several strategic areas. I'm going to focus almost exclusively on the second one shown on the slide, which is that we can make human antibody phage display libraries that are focused on difficult to drug classes of targets. And the first class of targets that we've gone after is the most widely validated but difficult to drug class of targets, G protein coupled receptors. So we now have 2 libraries. I'll talk about one of them in depth today, which is our motif guided GPCR antibody library. We've also created a whole series of other antibody phage display libraries that are more general use, both Fab, single chain, and VHH libraries, which I won't talk about today, but certainly please follow up with me if you're interested in those. And then we also have a platform for doing antibody optimization. So if we find an initial lead from our libraries, we can always make it better through our optimization platform. That I also can provide you information later if you're interested. And then finally, the fourth area that we're focused on is trying to extend the Twist SynBio offering. Again, Twist is a major player in the space to synthesize clonal genes, but we're trying to extend that into antibody production. So someday in the not too distant future, you'll not only be able to order an antibody gene, but also the antibody that's encoded from that gene from Twist. So again, our advantage that we see in this space is that we can make precisely defined libraries using the library platform I just talked about to make unprecedented, very high quality antibody phage display libraries that are devoid of any liability issues and match the natural human repertoire and also potentially contain sequence motifs, which is, again, as I said, is really key to the GPCR library that I'll talk about today.